I'm sorry, I think there was a technical glitch. So let's talk about today's session where we come together to discuss with our patients and eminent experts to discuss about cancer patients as they fight their battle against the disease. Our series is called Caring Conversations Empowering Lives for Cancer Care. In today's event, we will try to understand what is meant by real world data and the value it has to accelerate cancer research. Then we will hear from our distinguished guest, Dr. Shom Shubhronath, about the need for complete longitudinal data which can bring out insights from this clinical trial data. We would encourage you to ask questions to our experts by typing them in the comment section. We will take them up at the end of the session. Let's begin with today's session of Beat Cancer Facts. In today's session, I would like to explain more about what is meant by real world data. It is the healthcare information that comes from outside the clinical research setting. It can include data like electronic health records, that is, the patient's lab results, the treatment plans, the radiological images, etc. It can also be the data from patient and disease registries. It can be data from social media platforms where the patient community writes, blogs, twitters, etc. It can be patient reported outcomes that the patients experience the symptoms, etc. And it can also include information from wearable devices. So nowadays we all wear smartwatches and other such devices from which the healthcare related information can be collected. So RWD is paving the way to generate insights that can drive decisions in the life sciences and the healthcare industries. It can provide information about healthcare outcomes from diverse patient population, and that is why it's a very valuable source of data. This information from diverse patient population complements and expands on the information collected from traditional clinical trials. Additionally, it can help in validating the findings from the clinical trials and evaluate the effectiveness and safety of treatments, the products, the patient programs in the real world settings. Experts in the life sciences industry, including pharmaceutical and biotech companies, can use this information to support regulatory approvals, post approval validations of their products and interventions. RWD can also support research and development, including design of the clinical trial, the identification of unmet healthcare needs, and of patient subgroups that may benefit from the specific treatment or the intervention. All of this can help drive improvements in patient care, including earlier and more accurate diagnosis of cancer and better treatment options. There is a related term that is also used sometimes, which is the real world evidence or RWE. RW is nothing but the analysis that is done on RWD, and it provides clinical evidence about the usage and potential benefits or risk of the medical product. So RWD is answering the pertinent research question and analyzed on the real world data. Now, because the real world data is routinely collected outside of controlled study settings, it imposes some challenges, particularly with respect to the data quality, which may be inconsistent, inaccurate, or incomplete. The lack of standardization in data collection and coding across different healthcare systems and regions poses a challenges for RWD integration and analysis. There are also ethical and privacy concerns related to patient data collection and sharing, which are additional aspects when we are using real world data and generating real world evidence. To discuss more on the opportunities and challenges of real world data, we have with us today our distinguished guest, Dr. Shom Shubhronath. He is a highly accomplished researcher in the field of cancer, cell, and molecular biology, with extensive experience and wealth of knowledge in the field of oncology. He has worked in premier institutes in India and USA. He had a pivotal role as a scientist in the Basic and Translational Research Division at Saroj Gupta Cancer Center and Research Institute, Kolkata. Currently, he is the assistant professor in Institute of Health Sciences, Presidency University, and he is working on identifying novel mechanisms of resistance to frontline tyrosine kinase inhibitors in chronic myeloid leukemia patients. He has also worked extensively to decipher the chromosomal instability and aneuploidy in human breast cancer. Dr. Nath's professional journey not only deepens our understanding of the complexities of cancer biology, but also carries potential to accelerate advancements in diagnosis, treatment, and care for cancer patients. With this short introduction, I would like to welcome Dr. Shomshubrana to our event. We eagerly anticipate the insights and knowledge that you are poised to share with us today.
Thank you, Dr. Mandal, for this nice introduction.
Thank you. So, before we begin, can you please explain a little about your research, the insights that you are trying to gain, and what all data sets you are using?
Okay. So, as Dr. Mondal has explained, I am a cancer biologist actually. I deal with the cell and molecular biology of cancer cell, and I am specifically focusing on two fields of cancer. One is breast cancer, another is chronic myeloid leukemia. Now, breast cancer I selected because, as we all know, it is the most common cancer among women worldwide as well as in India. So, in the field of breast cancer, I'm trying to find out the molecular mechanism of a drug therapy that is specifically known as endocrine therapy. So, let me explain in a line that endocrine therapy is recommended in a breast cancer patient whenever the patient gets detected with hormone receptor positivity. So, whether a patient is hormone receptor positive, whenever the doctor prescribes endocrine therapy, but it has been found that in the course of treatment, many of patients just comes with the resistance against this therapy. So, I'm trying to find out the molecular mechanism behind this endocrine resistance. And for that, I need to know the scenario of endocrine resistance in our cohort, that is from the Indian population, as well as I need to validate this endocrine resistance from the global cohort. So that's why I am dealing with the real world data from the Indian scenario as well as from the global scenario. The second field of work is chronic myeloid leukemia. Here too, I am trying to find out a molecular mechanism that is working behind the drug resistance. Here the drug is tyrosine kinase inhibitor therapy. And for that too, I need to access the real world data globally as well as national at the national level, so that I can get the information in which ratio the patients are being diagnosed with the drug resistance and what is the outcome of this drug resistance. So yeah, for both of my studies, I'm dealing with the real world data.
Okay. So when you are collecting the patient data, we really want to understand how do you ensure the privacy and confidentiality of that data, because you are using it for research purpose. So how do you maintain that?
Yeah, that is a very valid point you raised, Dr. Mondal. So, whenever I go for collecting the patient sample, biospecimen as well as the patient data, the first step is to go through the institutional ethics committee. So, for each institute, there is a institutional ethics committee. The committee looks up for the ethical aspects of the of this study, and the committee ensures that the identity of each and every patient who got recruited in this study remains confidential, private, and anonymized. As a researcher, myself and my representative are provided the biospecimen and the patient data, but we generally don't get the patient information like his name or the contact address or the phone number, etc. So we only get the registration number of the patient, so that whenever we need to go for the follow-up information, we just check the registration number to know whether the patient is again registered or coming back to the hospital. So in this way, we from our side and the institute from their side are ensuring that the patient's identity are completely put under wrap, and that is not disclosed at all.
So the patients need not worry about being contacted or their information being used for any other purposes, right? Because you do not have it for your own use itself.
Yeah, so only the clinician acts as a liaison between the researcher and the patient, and we do not have the access to the patient until the clinician allows us to meet the patient directly.
So is there any need to meet the patient directly, like you mentioned?
Uh, this is needed when at the first round, when I am explaining the utility of this research to the patient or patient party, myself or my representative needs to meet the patient or patient party so that we can explain why we are asking the patient to join in this study. So that's why we need to meet the patient at least once at the beginning, when the patient will be enrolled for the study
So
or the
this is
patient take the decision.
so is this a part of their informed consent form then?
This is
They
the
need.
part of the exactly. Yeah.
Oh, so, can you tell us a little bit about the challenges that you have faced when you try to acquire the RWD data or manage it, and how that can be overcome?
Yeah, there are several challenges actually. I wanted to get this question so that I can explain. So if I want to summarize this part, I could find there are two, at least three challenges. So the first challenge is the uneven socio-economic status of our country or of our part. So in this condition, many patients opt out from the treatment after the initial round of surgery or initial round of treatment. So when a patient is opting out from the treatment or selecting another institute for the later phase of treatment, that we are not getting the follow-up information of the patient, because we do not have the patient's contact number or patient identity. We only have the registration ID for a particular hospital, that is here in my case Saroj Gupta Cancer Center. So whenever the patient is opting out from the treatment due to the socio-economic status, might be so we are losing the follow-up information. The second point I must say, there is the lack of awareness about keeping those real world data. We only know that these data are being recorded when the patient is meeting or explaining his or her condition to the clinician. But we often do not understand or do not know that these data are very crucial to extract the information about the patient's outcome, survival outcome, or the outcome from the treatment. And that makes the sometimes many a times the data is being stored only in the hospital desktop or in a shelf of a almirah in the hospital. It is sometimes it is lost, it is misplaced, or sometimes it is kept there only. There is a third point also. There
Not
is a high
digitized. It is not digitized, so it is
right
not easily
properly or
accessible. Yeah.
yeah. And if even if it is digitalized, the third point is that there is a huge population rush. So a clinician gets a very little amount of time to meet a patient and for a prolonged period, so it might lead to the lesser recording. Means a patient may have several things to say, but there are a long queue of the patient and the clinician has to attend all of them. In this condition, the data recording may not be complete or adequate. And when we are trying to extrapolate this data, we are getting, say, inadequate information or lesser information than we should get or that should be there.
So the data is not uniform, it is not complete. You don't get it for
Not
extended period of times for the follow-ups.
exactly. Yeah.
Okay. In spite of these challenges, can you provide few examples of specific insights or breakthroughs that have been possible because you could analyze this patient data?
Oh, definitely. There are several insights I could get or the data analysis could give me. So, like for one example, I can cite. I have heard that the prevalence of triple negative breast cancer it is a type of breast cancer and it is the deadliest form of breast cancer is much more prevalent in the Asian context. And when we mined the data from our hospital, that is Sarajgupta Cancer Center, we could find that yes, it is the case. Here too, the prevalence of triple negative breast cancer is much more higher. Also, I could find that there are more younger onsets of the disease. That is, cases who are getting diagnosed below 40 years of their age are much more higher and they are showing much poorer outcome than the older cases. And that is the Asian scenario. So, from the eastern Indian part of India, we could find that the Asian scenario is there also.
Okay. So that means that whatever has been found out globally can also be corroborated with Indian population, and that is
Yeah,
why this real world patient data really helped you.
really
So
helpful. Yeah.
you mean then it removes the potential biases in the data, or you need to deal with potential biases in the data, right? Because you talked about Asian population, but there are other changes in terms of ethnicity, diversity. So then the research has to represent the entire population. So how do you
It
deal with these potential biases?
uh, there are a few potential biases that I found when I started because I was working from one particular hospital that is Sarojgupta Cancer Center and that caters the Eastern Indian population that is situated in Kolkata. So I could only get patients from Eastern Indian population including the Bangladesh. So I was missing the patient from the other part of country. So my analysis was skewed because it was only comprising this part of the country. For that, I had to collaborate with some other institute. So I reached out. I contacted some other institute from different parts of country, like from the southern part, northern part, as well as from the western part. And in this way, I could get the pan-Indian scenario of the particular thing that I am looking for. So in this way, I could like go beyond the biases of my data analysis.
Okay, that's really important to understand, because that gives an opportunity to researchers across the globe, across the country, to collaborate and then give us an insight which is more comprehensive in terms of representation. So I think that's a wonderful thing to do. Can you please also elaborate on the types of analytical techniques that you use to extract meaningful information?
So for my study so far, I used say three or four types of analytical procedure. The first and foremost is that was the qualitative analysis where I just extrapolate the patient information that different clinical pathological parameter of the disease and I wanted to find what are the prevalence of different type of disease condition in my cohort. there was diagnostic analytics also, where I wanted to know, or I'm trying to know, what is the outcome of a particular treatment, whether the patient is like responding to the treatment well, or it is the treatment is not working at all. So from that data I am just extrapolating, and I'm trying to find out the association analysis with a disease outcome and the treatment modality. And the third thing what I am doing, that is the most crucial, I think, that is the survival analysis. So I am performing and I am checking the longitudinal assessment of the my cohort, how long they are behaving well or staying disease free, and that is the disease free survival, and how long their life expectancy is going really well. So in this way I am performing the survival analysis using Kaplan-Meier plots or other Cox regression analysis. So a few statistical analysis I'm incorporating.
Yeah, that's important to know. And I'm guessing that the hospitals from where you are getting the data from, so there are other medical professionals with whom you can also collaborate, right? If they are in the, so that these research findings can be translated into the clinical practice with the help of them.
Yes, definitely. As I mentioned before, a clinician can only be a liaison so far between the researcher and the patient or patient party. So, the first step is to collaborate or to find out one clinical collaborator who might be a surgeon if I am looking for the biospecimen, who might be a chemotherapist or the radiotherapist while I am dealing with the longitudinal assessment of the patient, or some other medical professional, so that I can get the clinical data, the validated, clinically validated information from a registered, certified clinician only. So this kind of collaboration with the clinician is the utmost need to conduct any clinical research, and so that upon doing this clinical research I can translate or I can inform the clinician, yes, now it is ready for a translational researching. So a clinician can start the clinical trial from there on.
Yeah, that's, so that is bringing one of the stakeholder into the loop. But the other hand, there are other stakeholders like the patients themselves. Are they aware of the potential of this data, that it can really advance cancer research? Are these insights shared with them, whatever insights that you get from your studies?
They must know or they should know. As I again mentioned before, that at the initial round, when I am recruiting a patient for my study, I try to explain, myself or my representative try to explain the utility of this research and why this research is important, even not for himself and or herself, this is for the entire nation. So why the data is getting the data performing this clinical research is important, that must be explained to the patient or patient party. But there is the another problem layer of complexity, because as I mentioned, there is a heavy rush, the patient needs to attend several clinics, several diagnostic steps at the same time, and the patient might not have enough time. And the most important thing, at the time when I am discussing this to the patient, that patient is going through enormous trauma, not only the patient and his or her family. So it might not be sufficient to understand all the thing I am explaining at one go. So for that, we need a few more sessions to explain the utility of this research.
So, just a follow up to this, is the posthumous data of the patient helpful? Because that would also be real world data, but the patient may not be alive to give their consent. So, is that kind of data also used?
Yes, and that we call as retrospective cohort. That is the cases who got registered in the hospital for a long time back, say 5 years back or 6 years back, and the patient had followed up for a period of time, and now the patient is not reporting to the hospital. Might be for several reasons, but that is the retrospective data. So from that data, we could also extrapolate a few information. But if we want to do some real time analysis, like the genomic analysis or the proteomic analysis, we need to get the retrospective, prospective cohort where the patient is, I am meeting with the patient and I'm getting the biospecimen. So yeah, the retrospective data is useful too for several things, but prospective is most
Because you
influential.
can get the biospecimens from them to do the live data
Yeah, yeah,
analysis.
yes.
Okay, understood. So how would you say would be your like advice to the patients, or how can we encourage them to contribute their data to medical research? Uh, there may be many people who are not even aware that they can contribute, right? So what would be the best platform to do that?
Uh, the, for the based platform, I will seek the advice or the help of a caregiver. And that is the most important part, because myself being a researcher is getting very limited access to meet the patient. And the reason is clinical as well as ethical. A clinician too, though he or she may have access, but the clinician has limited time because that clinician is attending more than one patient in a short period of time. So only a caregiver who is dealing with a patient is spending more time with a patient or patient party than a clinician and a researcher. So if we can train a caregiver properly, and if a caregiver can act as a liaison between the researcher and the patient, and then only a caregiver can transmit the information to a patient or patient party about the utility of the research. And in this way, it can spread in a word of mouth fashion, or it could spread in a hospital or in a diagnostic setup. So the role of a caregiver is very much crucial here to encourage each and every patient the caregiver is attending, or the patient party the caregiver is attending to. So in this way, I think only we can encourage more patient and patient parties to be willing to participate in the longitudinal assessment of the real world data.
So you mean to say there should be a proper awareness about these themes, that these people can actually help us to help them, right? So that is the message that needs to go across.
Yes, yeah. There is a pivotal role for the caregiver in this
Yes.
platform to act as a liaison. Without their help, I think we couldn't make that awareness possible.
Yes, certainly. So, thank you, Dr. Nath, once again for sharing with us your expertise and insights. But unfortunately, we have limited time and we cannot continue this discussion today. But we really appreciate your time and valuable contribution to today's event. Thank you so much. It was a pleasure having you here.
Thank you, Dr. Mohan.
Thank you. With this, we come to an end to today's event. Thank you all for taking part in this engaging session. We look forward to seeing you again next week, when we will have with us Dr. Oliver Fante, who will enlighten us with the current advances in healthcare and possible early detection of cancer using artificial intelligence technology, the buzzword for today's time. Thank you. Have a great day.